Okay, it is 4 minutes past the hour, so I guess we can make a start. So good afternoon, evening, morning, everybody, and welcome to the Palaeoverse Lecture Series. This lecture series is focused on software development and methodological approaches in paleontology, with the aim of promoting the use of new tools in the field and to give researchers a platform to showcase their work. With this series, we hope to shake up the usual style of lecture by having a mixed format with time dedicated to the presentation, demonstration, and discussion of the software or method presented. Typically, these sessions will run for about 50 minutes in total, and we will run this series throughout the year, generally on the last Thursday of each month, unless they overlap with holidays. And we have a lot of holidays coming up in the next quarter, so keep an eye on those. If you are interested in presenting a new method or software you have been developing or implementing, then please feel free to reach out to us. We are always happy to hear from the community. So before moving on, just a few quick introductions. My name is Bethany Allen, and with me today I have Erin Dillon, Lewis Jones, Will Goethe, and Harry Drage. The five of us are on the core team of Palaeoverse, an organization aiming to bring the paleobiological community together to share resources, reach agreed standards, and improve reproducibility in paleobiological research. If you want to know more about the Palaeoverse, please check out our website and GitHub organization, which we will post links to in the chat. So that's all from me for now. Erin will now give you some helpful tips and instructions for this webinar, and Lewis will introduce this month's speaker. Thank you very much, and please enjoy the lecture. Over to you, Erin.
Awesome. Thank you, Bethany. So as Bethany mentioned, the format of this seminar will consist of a talk by our speaker, followed by about 10 to 15 minutes at the end, which will be dedicated to Q&A and discussion. So my fellow hosts and I will help field your questions and moderate this discussion. Please note that the seminar will be recorded and may be made available on the Palaeoverse YouTube channel. However, we will stop the recording before the Q&A period in case you would prefer not to be recorded. So before we get started, I'd like to quickly point out a couple of useful Zoom controls at the bottom of your screen. You'll find a Q&A button. So you can use this to submit your questions to our speaker, and during the Q&A session, we'll either read out your question or invite you to turn on your microphone in case you'd like to ask the speaker directly. As Oskar goes through, he may also be able to answer some of your questions if there's any burning questions that are on your mind. There's also a raise hand button. So alternatively, you can raise your hand and we'll either call on you to invite you to turn on your microphone so you can ask your question. There's a chat function. So currently you can only chat with the host in case of technical difficulties, but we'll allow you to chat with other attendees during the Q&A period. And then there's a closed caption button that you can turn on and view the transcript at any time. So last, I'd like to highlight a couple of ground rules. Our aim is to cultivate an open, respectful, and inclusive environment during this lecture series where we can all learn from each other. So with this in mind, we ask that you please be respectful and kind when engaging with the speaker and fellow attendees, particularly when asking questions and interacting with others in the chat. Any form of harassment or inappropriate behavior will be grounds for removal from the webinar platform. So for more information, please refer to the Palaeoverse Code of Conduct, which can be found on our website, and I believe we'll just put a link in the chat. So with that, I will pass it over to Lewis, who will be introducing today's speaker. On to you.
Thanks, Bethany and Erin, for getting us all up to speed on how the lecture series works. So today, I've got the great honor of introducing our speaker, Dr. Oskar Hagen. Oskar is an evolutionary biologist and environmental scientist whose research explores one of the most fascinating questions in science: how biodiversity emerges and evolves across time, space, and complexity. Now, Oskar leads the computational integrative biodiversity research group at the Center for Critical Computational Studies at the Goethe University, Frankfurt, and is also affiliated with the Senckenberg Research Institute. His research integrates diverse disciplines from ecology, evolution, and biogeography, all the way up to geology and climatology, with the aim of kind of better understanding the processes that have shaped life on Earth. And by developing and applying spatially explicit mechanistic ecoevolution models, or MEMES for short, Oskar investigates how ecological and evolutionary and deep time environmental factors, including plate tectonics, influence biodiversity patterns. Now, through this integrative and collaborative approach, Oskar's work not only deepens our understanding of biodiversity, but also exemplifies the power of connecting diverse minds and methods to study life in all its complexity. So with that, please join me in welcoming Dr. Oskar Hagen to our virtual stage. Over to you, Oskar.
Thank you, Louis. Yeah, thanks for the introduction, and thank you, everybody, for the invitation. Let me share. So, is it working?
It's just yep, looks good.
Cool. All right. So today, topic is, as Louis very nicely already introduced, is on mechanistic biodiversity modeling, and especially the second part on trying to span multiple spatial and temporal scales. I normally like to give a talk and interact with questions, but if you have a question and you come up with a question while I'm presenting, just write it down on the chat, and maybe you don't miss the inertia to just ask the question. And for today's talk, I would use the latitudinal and the longitudinal biodiversity patterns as a guiding thread. And first, I will set the stage with some motivation and context, especially how gen3sis, this engine, came to be. And then I will dive on the methods and hands-on part, which is today's main focus. And finally, I will wrap up with some case studies and giving a very brief outlook. So biodiversity is unevenly distributed around the globe. As you can see in this map of richness of vascular plant species, with some places with very few species and other places with many species. And this leads to very characteristic patterns, as many of you may know, such as, for example, the latitudinal diversity gradient, which is the increase of diversity towards the poles, and the longitudinal gradient, or the pantropical diversity disparity, which is an observation that was made already in 1973 by Richards regarding the Afrotropics and its lower tropical diversity compared to the Neotropics and the Indo-Malayan tropics. So the questions I'm interested on are really broad, and they go roughly on why are there such distinct patterns, and also what are the processes that shape or shaped biodiversity, right? Really broad questions. And to answer these questions, there are many existing theories and hypotheses. But these theories, sometimes they're loosely defined, or they still have very little agreement, or they're mostly verbal, or they just tackle a partial component of this complex system. So here, as an example, I'm showing you some 6 such theories that are proposed to explain the latitudinal diversity gradient. And when before I developed gen3sis, the idea to tackle each of these single hypotheses was very overwhelming, because there is still a lot of uncertainty on how they are combined, and also how these hypotheses are to be implemented in a broader context. So what I like to do on the research is just basically step back and look at these theories and hypotheses, and when I do this, I can spot cartoons of 2 main perspectives, right? So some of these theories are mostly ecological and others are mostly evolutionary. So what I mean by this is, when you think about the ecological perspectives, they are mostly spatial, right? And then we consider current abiotic or biotic conditions or species interactions. And we will see, for example, species response along an environmental gradient. While on the other hand, at the evolutionary perspective, we have a much stronger temporal perspective, and not so much spatial component. And from this perspective here, we will think of processes such as macro evolutionary processes like speciation and extinction events. And we know that on top of this, we have a very dynamic landscape, a scenario where this historically diverse climate and geography, they lead to very different habitat regimes. So there is a very strong need of interdisciplinarity here so that we can understand how biodiversity patterns are formed to ultimately predict how they will respond to global change, right? Although this predictive part is really a long-term goal and probably not that close. So we know that biodiversity patterns emerge from multiple processes, such as species interactions with biotic and abiotic conditions, species speciation and extinction events, as well as species adaptations to past environmental conditions that changed over time, such as continental drifts, mountain uplifts, and changes in temperature and aridity. And these processes, they interact in very complex ways that we have to consider in order to move beyond statistical approaches. And this knowledge is accumulated since the early 1800s with figures such as Wewel proposing an inclusion of time dimension, or Charles Lyell, or even Alexander von Humboldt, that recognized this ever-changing world and basically brought natural history closer to natural philosophy, right? And these thinkers, they all already started influencing the ideas of Darwin that I put here as a 7 years old young naturalist. So although many smart people have been thinking about this for a long time, nature nonlinearity, complex memories, as well as the very impossibility of doing experiments at deep time and large scales adds to the challenge of finding causality. And this is a very strong contrast to, for example, physical laws such as the Newton third law. But luckily, we can use computers to assist us solving these assumed interactions and solving these theories or propositions that we might have and enhance our intuition. So when I'm talking about computers models, I'm talking about a very specific category. So now I'm narrowing down to specific methods. And as Louis presented already, I call them memes, which are standing for population-based, spatially explicit, mechanistic ecoevolutionary models. It's a mouthful, but I will go through the main ideas. And if you would like to see a review on these methods and some perspectives, you could check this publication on coupling ecoevolutionary mechanisms with deep time. So basically, memes, or this class of models, they model individuals implicitly. So the explicit unit are populations. And models, they explicitly apply ecoevolutionary components that are acting at the same spatial and temporal scale at this population level. So this is a conceptual shift from considering not individuals, but populations as the unit of analysis. And this is not something new, because Darwin even proposed on his search for a common history of all creatures through the changes occurring between and within populations. But when we assume that populations are the unit of our analysis and the unit of these models, we have some advantages, and mostly they can circumvent the complexity and the level of details we would need to run individual-based models, right? So you can imagine having to consider individual interactions, and once you're modeling individuals, you are stuck to the generation time. The other advantage is also that we can simplify at some level this complexity or this unknown and model this population dynamics. By having this and being able to cover very deep and large spatial and temporal scales, we basically can study spatial effects such as geometry, and we can also cover a multitude of spatial and temporal scales, because the population as the unit of analysis, they can cope with changes in temporal and spatial scales. And I will try to illustrate why this advantages through my talk, but intuitively, you would want to build a single or a general framework where researchers or people studying different scales and temporal scales and spatial scales can condense and can synthesize their knowledge in an exchangeable way, right? So within these kind of categories of population-based mechanistic models, I'm going to present one tool, which is the one I developed when I was kind of tackling these questions I presented. And it stands for General Engine for Ecoevolutionary Simulations. And I decided to call it an engine because, like a gaming engine, for example, the CryEngine or Unreal, I don't know if you're familiar with some of these tools, but these gaming engines, they basically allow the users to create their virtual world where they're gaming, right? So I don't see gen3sis as a model per se, but as an engine. And this engine is basically just helping users to create these different worlds, these different models, while keeping a certain level of formality and rigor, so that once you build and create some ecoevolutionary rules that you're interested, all of your secondary assumptions are also very explicit and are also very easy to understand and follow. So this engine has some components in C++, which is mostly the small critical part, which is not very flexible, but most of it is programmed in R. And it was a deliberate choice because R is a language that is very accessible to different disciplines, life sciences, but also geology, climatology, and etc. And it's also a very accessible language, right? So this is all under GPL3 license, and it's available also as R package. So the idea of gen3sis, or this tool, is really to formalize this very different biodiversity theories. And by formalizing, I mean creating this kind of structure where you take a knowledge that is verbal or mathematical or fragmented, and you can put it in a larger context where space, time, landscapes, ecological and evolutionary components have to be considered. Or if they're not considered, they have to be explicitly mentioned. Okay, so beyond formalizing, this tool or this engine allow researchers to create and to test these different hypotheses, to do hindcasts, but also do future projections. And ultimately, it will allow us to build counterfactuals, to play with assumptions and with our reconstructions, and quantify and link these very different distinct processes that are leading to the emergence of biodiversity, right? So the idea is really to explore spatially and temporally how biodiversity patterns emerge. Okay, so let's jump straight into how this engine works. So as you can see here, the engine is just the middle component of a model, which consists of an input that are a dynamic landscape and a configuration file informing the initial conditions and the ecoevolutionary rules. We're going to look at these both components very soon in more details. So here, the configuration you can imagine is a file that is just saying under which conditions, what were the initial conditions, but also what are the ecoevolutionary rules for this world. These are then fed into the main engine that will compute species abundances, distributions, population divergences, phylogenies, and trait values. And this leads to multiple emergent outputs that can then be compared to empirical data. So here's just a few examples of the outputs that can be generated using gen3sis. So you have present distributions, past distributions, you have phylogenies, you have ecological traits, and a multitude of further analysis that you could do combining this biodiversity dimensions. Okay, so here an example of such a landscape that we use. Here we have world map of the Earth surface temperature and aridities for the last 200 million years ago, and this was reconstructed using plate tectonic movements and topographical changes, lithological information, stable isotopes, and fossil data. So using to simplify this landscape, now I would like to show a very small cartoon on how the engine works. And for this, I'm using a very simple landscape here composed of some highlands, some lowlands, and a small river. I hope you can see my mouse here. So basically, the idea is that these landscapes, they are changing in discrete time. And every time we have a change in this landscape, which we have a process of landscape and dynamics, then we trigger a sequence of 3 or 4 main processes within the engine. And to show now these processes, I would use a single population of, a single species of a brown fox. Okay, so we start with the speciation, where we basically account for gene flow between populations. And here we see a gene flow happening between these connected populations here, but there is gene flow interruption between these 2 populations. And this can happen because of landscape properties, but also depending on the species properties, such as, for example, dispersal abilities. And in this case here, the isolated populations, they start accumulating a genetic distance, which can decrease if they get, again, in secondary contact. But basically, if you keep them isolated for long enough, you would reach reproductive isolation, and you have a speciation event. So in this case here, there was no speciation event, but there was accumulation of genetic distance between these populations. Then these populations, they would disperse in the landscape. So we see here this foxes going through the landscape. Once they disperse, they can have their traits mutating. So here we see a white fox appearing on the highlands and a red fox appearing on the lowlands. And then once we enter the ecological module, we then go for every single site and we calculate the abundances or the presences of these populations, possibly considering biotic or abiotic conditions, and possibly leading to extinction. So here, for example, this white fox was suitable for the highlands, while this red fox was not suitable. And I think the point here I would like to stress is that each of these single components, they are very flexible, right? So these are not hard-coded. So these components are informed by the configuration file, which takes a very different set of functions. So for example, we could consider a very simple neutral model, or we could consider different biotic filters or abiotic filters or species interactions. On speciation, for example, we could use environmental conditions for the simulation of divergence or any other more sophisticated processes or theories. On the dispersal, for example, we could model something much more deterministic. We could model something that is more probabilistic through a probability density function. And on the traits evolution, we can also consider multiple different processes as well. So now I would like to jump to the tool itself so that this is a little bit clearer. And what is important to note, and I will put a link on the methods which make it a little bit easier to follow, is that speciation, dispersal, and evolution, they are happening basically through every species. So the simulation within each time step, then it's looping for each species. But on the ecology, then we solve each site, right? So we go for each site and we look at the local conditions, biotic and abiotic, and calculate these abundances. So let's make this a little bit more concrete. I already installed gen3sis, which is on CRAN, but here I will just run some libraries. This is the library of the gen3sis. Then we have Terra, the here package, just to help me with the links. And here I'm loading ape to work with some phylogenies. So first thing, I'm just putting here question mark on gen3sis. You'll see here's the help, the first page. And if I just take here this first example, we can already run a very simple simulation. So this, you should have this already ready when you run. So this is just a very rough crop on the at 4 degrees. And I'm just using readily available configuration files and a landscape file. But I prepared something a little bit more realistic for today's presentation. So first, I'm printing here the gen3sis version. You see we are at the version 1.6.0. It's a race to keep things on CRAN, but it's there since I think 2 weeks. Here, I'm basically taking the data path where I have my landscape and my configuration file. So now I'm leaving RStudio and I would like to show you these files, so how these things look like, right? So remember, if you install the package, you already see this kind of very small and simple landscapes and configuration files. But here I prepared 2 landscapes. One is South America. So in South America, I have here input ready. So this is basically an input landscape. There is this object landscapes.rds, which contains this environmental variables and the sequence of events. Here, specifically, we start 65 million years ago, and we do 1 million years jumps. So right, time steps of 1 million years. This is very rough. I wouldn't do this scientifically, but just to cover a very broad scale and to save a little bit space, I chose to do this. And here you see we have a distance full and a distance local. So basically, you only need one of those 2. And the distances are just information, a pre-computed information that is given the resistance, or basically the distances between the sites in this landscape. And because this landscape is dynamic and the sites are changing because we have plate tectonics, I have this already distance pre-calculated. So this is a little bit a hack to make things run fast. But if you have limits on space, on storage space, you would like to compile your landscape using local distances. This means that I just keep the neighbor resistance. And once you start the simulation, gen3sis will then compute this full-blown full distances, right? So if you have storage space, I would definitely go for distances full, because in this way your simulations are just going to be much faster. Okay, So here you can see this landscape, and now I would like to go back to our studio, and we have the path to our landscape. And here I'm just reading this landscapes.rds object. Okay, so you see that this is a list, and this list has a temperature, aridity, and area. So landscapes, they can have as many environmental variables as one would like to, right? So this is just one example, but if you would have solar radiation or elevation or whatever environmental variables you have, you can put it in here. Only important is that for every site, if you have one of these environmental variables, you should have other environmental variables there as well, right? You cannot have temperature information, but then you don't have kind of aridity. And here I'm just showing, so these are like, basically we have, in this landscape, we have 5,751 lines and 68 columns. And basically, the first two columns are just the X and the Y. And then we have the temperature variable. For example, here I'm accessing the temperature list. And we have the first 10 lines and the first 10 rows. And you see the present, and then we go back on time. So here I would just like to plot this very first slice. Okay, so I'm using, oh, this is a very old raster from XY. This is a raster package thing, but we could do this also with Terra. But basically I'm plotting here this temperature for the present, and here I'm plotting temperatures and the South America map at 65 million years ago. Try to make it a little bit bigger so it's nicer. And here I'm just plotting an overlay. So you can see 65 million years ago in red, and you see how the landmass of South America moved. All right, so this is the configuration file. And now, what else we need? We need a configuration file, right? So the configuration file, you can create input config. So you can create a configuration file from zero, but I really recommend for you to start with something that exists already and build on top of it. So for our first simulation, I would like to use this config South America observer. So this is a file. I'm going back to the Explorer. So I just store this file here, config South America observer. And this file, we will have some general settings. For example, we have here a random seed. This can be NA. This would mean it will always have a different random seed when it's run. But what I like doing, especially when I'm designing this ecoevolutionary config, I like to set up a seed. So this is just today's date. And I just do this because it's easier to follow if there are mistakes, and there will be mistakes, because as gen3sis is very flexible in general, the learning curve is very steep. Great, so here we have a random seed. Here we have a starting time, so I can decide if I start like at the time type 40, or I can just put here NA, and then I start with the simulation at the oldest available time step. This is the end time, so when it will stop. So if I just leave NA, this is gonna go to 0. I'm just gonna put here 40, so this is not taking forever. Here we have the maximum number of species and the maximum number of coexisting species. And these are just stopping criteria, okay? So this can be infinite. What happens sometimes, you want to have some embedded stopping criteria just to stop simulations that are going wild, or there's a lot of turnover, for example. And here is the trait names. So this is already the first part where you start seeing how flexible this can be. So imagine you would like to model biomass. Then you would just add biomass here. And on the other steps within this landscape, you would have, within this configuration file, you would have to think really hard how biomass would interact with processes such as speciation or dispersal or evolution or ecology. Okay, so in this case here, it's a very simple config where we have species having optimum temperature and a dispersal ability. Okay, so I'll just open it. Oh, no, it's not so nice. So maybe the first function is the end of time step observer. And this is a function that is called at the end of every time step, or depending how you set up your simulation. But here I'm basically making plots, right? So if I would like to, for example, save the species objects or save a phylogeny or plot the richness in this landscape. So there are a bunch of suitability functions. So here you could also, you can use this to plot, or you can use this to pre-compute, or while you're simulating, just store specific data or create an observer data on some preservation functions, for example. So for the initialization, the initial abundance is basically the abundance value that you have for a population when it's created. And here we are creating an ancestor species. So the create ancestor species would be the first call, which would set really what are the initial points or the condition for this landscape. And before I run the simulation, I would just like to put a browser function here. If you don't know, browser would be your best friend or one of your best friends if you're modeling on gen3sis, because since these components are within a configuration file, if you have a browser, what it makes is once you reach that browser statement, R is going to throw you the environment that you are in. So you'll be able to see what is happening in there. And then from there, you can play and debug or create your function. So here, I'm just putting a browser function, right? I'm saving this, and now I'm going to run the simulation. So sim run simulation. And here I have the configuration file pointing to the South America observer. I have the landscape. I'm calling all time steps the observer function. And voila, I started running, and then I get already thrown to my first browser. And you see here, I'm inside the create ancestor species. So once we are here, what this is doing, in this case, I won't be able to go too much into details, but here I'm basically taking a certain range. I'm taking the coordinates because I have the landscape and the configuration objects, right? So I'm setting this range. Once I have this range, I then create 10 random species in this landscape, okay? And when I'm creating this random species, I'm using here this create species, and then I'm setting up. So each population, so there are 10 random species, one population for each species, thrown randomly in South America or within my range. And these populations, they have their optimum temperature range exactly the same as the temperature range of the cell, right? The environmental temperature, so they're optimally adapted, and they have a dispersal ability of 1. Okay, so what this means, we will see later. So this is one example. But I could also, so let me run this. So I'm going to stop, I remove this browser, I'm going to save this config file, and then I'm going to run the simulation. So here we see already, we started. What we are plotting here are the species richness. And so the alpha diversity, you see how they're accumulating through time, and you keep track of how many species there are. I'm just going to stop this simulation here. And now I'm just going to go back to the first time step. So you see more or less we had this kind of random species here. It's the first time they already started dispersing. So now what I would like to do is I would erase this first variant that I created, and I would enable this second variant, so variant B, which is just creating one single species spread everywhere with a temperature of 28 degrees and the same dispersal, right? And I'm just setting a plot species presence here so that we can see where species number 1 is. So if I save this and I run, you see that the starting condition is different, right? We have a species everywhere, and then we already see accumulation of species here, and so on. Okay, so you can already see how we can interact with these two things. Let me go back to variant A. I think I was too excited to present this and I put too many variants here. But let's go through this. So the divergence threshold is basically playing together with the get divergence factor. So here it's a very simple, it's like a neutral case. So if populations are isolated, they will gain a marker of 1. And you can think of this like an FST value. And this is basically saying if populations have 2 or more distance, then they will become 2 different species. Since we are getting a marker of 1 for every time step, and we are changing the landscape every 1 million year, having a divergence factor of divergence threshold of 2 means that we have speciation event if populations are isolated for at least 2 million years. Okay, on the evolution, what we are doing is we are taking, for every species, we are getting information of the clusters, so which populations are connected, and then we can homogenize these traits and we can create new traits variants. And then on the ecology, again, we go for each single site. So basically, we're going to get all of the species, right? So all of the populations of the species that are on this site. We get their abundances, we get their traits, we get the environmental values of the landscape of that site. And you can do multiple things. So here is a case, is an example, a variant where I put some carrying capacity and I'm changing these abundances. And here is a variant where I use a very simple function, which is declared here as my function. So this is just to show you can add your own functions and complexify this config. And here it's just a Gaussian curve for niche. So basically, this is just telling, okay, if we have an X, like a gradient from -20 to 50 degrees, and we want to plot this whole gradient, you will see basically the abundance response for this within this temperature. So here, what we're doing is we're basically taking the response of this species in this landscape. All right. So here we will run this one simulation, and you can see that they're accumulating diversity. I won't stop for the sake of time. I will stop here because, yeah, it's quite fast, but oh, of course. Okay. Anyways, let's wait for this to finish. So once this simulation is run, what we have is an REN object that contains a summary of the simulation, but at the same time, we are storing things in this output folder. So in this output folder here, once the simulation is finished, we have basically the initial ranges, the initial richness. We have also phylogenies. And depending on the plots we're triggering, we have also all of these plots and this, and we have also a copy of the configuration file for reproducibility. So here we can either call direct this object, which is the scene, or we can just read this as gen3sis.rds, which is kind of a very small summary of the simulation, right? But it's important to know that all of this trait data, phylogenies, abundances, they're all stored locally. Okay. So while we're letting this config South America run, I would just like to show you another config that I prepared. And this one is running for the entire planet. And here, at the end of the time step observer, what I'm doing for fun, I'm saving the species and I'm saving the phylogeny because I want to plot the along this richness map. Okay. And then I'm creating, based on the same way, I'm just creating an ancestor species everywhere that is adapted at 28. And here I have viable, so it's a probability density function describing the dispersal. And this can be, for example, also an exponential, or you can use a completely deterministic function. Diversion threshold here, I just put it much higher. And for the ecology, I basically use the same example as before. So we have the same function I presented before, but I just added an additional function, which is some stochasticity on top of these abundances. And then I'm plotting the abundances. So, okay. So we're still simulating. So we see we started this one with 10 species and we are ending it with 45, 46 probably. Great. So if we look at the scene, we will have like a summary, we will have a flag. So if a simulation fail, you would get a flag. And this is important because what you want to do is simulate a bunch of things on the cluster. And here we have some convenience functions. So where you can plot, for example, the richness through time, speciation and extinction events. And here you can plot, for example, the phylogeny, and you see we started with the 10 species. And here I would just like to run this global example. So, and I also even added, once there is a speciation event, I added a centroid, which is a square. So once we have more than one species, we will see a phylogeny alongside this plot. Okay, I think for the sake of time, I will return to the presentation. Yeah, you can see here, right? So we had already a speciation event, and you see I also added a higher verbose here. If you go on verbose 3, you'll see much more details. Okay, so back to the presentation very shortly. This is the presentation where the article where I present these methods. And here we looked at latitudinal diversity gradient. But really the take-home message here is that you can explore multiple reconstructions, and you should, and also explore multiple configuration files. And through this interactive modeling cycle, which is not that trivial because we are not overfitting, we're not just looking at one single matrix of biodiversity. We're looking at multiple emergent properties within one considering single specific configuration files. So in this case here, we show that a carrying capacity on the area and the environment leads to kind of the reproduction of these multiple emergent properties, implying some ecological limits. And once we use this for the longitudinal diversity gradient, where we look at this pantropical diversity disparity, which we see here is very striking for palms, but also reported and confirmed with our data for other groups. there are many suggested hypotheses that it could be either related to past environmental conditions or present environmental conditions or lower extinction and higher extinctions. And as a take-home message here, we found out that really considering these past climatic variations are crucial. And when we look at the best matching simulations, we see that the Afrotropics had at the same time a lower speciation and a higher extinction, right? And this leads to insight and hypothesizing, okay, the Afrotropics, there was more extreme cooling and aridification with less environmental heterogeneity. But now it comes the interesting part, right? Because then using these models, they're not just the final say on what happened, but they're just a tool for you to start investigating and creating an intuition. So basically here we see, okay, there's much less environmental historic heterogeneity, less topographic change and topographic complexity, because the African plate, like the African continent, is just right in the middle of the African plate. So what we can do then is like go back, take these landscapes and basically erase the Andes or reduce environmental heterogeneity or island dynamics in the Himalayan tropics, or remove the aridity constraint on the African continent. Okay, so these are just a few examples. Here are just some further applications ranging from populations of a single species at a much shorter time scale, or with aquatic species, or even studying very large scale iconic biodiversity patterns. So to sum up and to finish it up, I would like to share with you the recent dimensions research I did of the research using and citing gen3sis. So I was very pleased to see that it's not only been used into biological science, but also in environmental sciences. And really the main goal of this engine is really to formalize, simplify, and connect. I would like to shamelessly promote a postdoc position that my group is open up. It's the first position. The deadline is the 12th of November. So if you like this and would like to play with these things, and not necessarily only with gen3sis, but these ideas of connecting multiple dots and exploring the unknown, please consider applying. And to finish up, I believe that advancing science spins currently really around the impacts of the disciplinary fragmentation. And for this, I would like to cite this Baleati study where they modeled scientists as agents in an agent-based simulation tool. And each scientist was impacted by signals received from the correct answer to the questions they pose, but also they're influenced by peer influence and also noise. And with the simulations, they were able to show that disciplinary fragmentation critically limits scientific progress, while limits in scientific progress do not necessarily lead to a disciplinary fragmentation. And this is a very clear message for me that progress is really made through social interactions and peer disagreement. And in the same lines as the enigma of reason, where it's discussed that logic and reason is something that is a social property. So it's an emergent property when different individuals with their own instincts and single ideas come in agreement or try to explain and debate their single points of view. And in light of this, I believe that tools that improve generality, flexibility, and accessibility. So let's say you take a study, you disagree with some secondary assumptions, or you want to test if things were done properly, or you can easily go there. You can understand the code. You don't have to change something hard-coded for a very specific question, but you have there already underlying base that makes things at least comparable and testable. And with this, I believe we can move towards a higher intuition, but also higher quantification, reproducibility, and communication science. So here I listed just 3 links. Maybe I will put it on share with the organizers so that they can put the links on the descriptions. But these are just very useful tools for you to tutorials or workshops that are organized on GitHub, or this is like a repository of a recent publication, so that you can really follow what was done and play with the configuration files. So I'd like to thank all of these institutions, also everybody from Palaeoverse for this invitation, and looking forward to your questions. Thank you for your attention.